Hello everyone, welcome to my channel Dr. TAPATI'S presentation. Today topic of the presentation is reactive oxygen species or ROS. In this video mainly I am going to discuss about sources of ROS, that means internal and external sources, defense against ROS, that means enzymatic and non-enzymatic defense systems, roles of ROS, that means constructive and destructive roles of ROS. Then finally you will get to know various diseases caused by ROS. This video may be quite lengthy, but kindly stay with me till end of this video because you will get to know important informations regarding ROS. First of all, know what is reactive oxygen species or ROS. ROS is an umbrella term of a group of derivatives of molecular oxygen which are very unstable and can easily react with other biomolecules in a cell. Aerobic life is completely dependent on oxygen, but some amount of inhaled oxygen is transformed to reactive oxygen species. Therefore, reactive oxygen species are natural byproducts of cellular oxidative metabolism produced as common outcome of normal cellular metabolism of oxygen. Biologically significant ROS elements can be categorized into two groups, free radicals and non-free radicals. Examples of free radicals are superoxide anion, singlet oxygen, hydroxyl radical, hydroperoxyl radical, peroxyl radical, alkoxyl radical, carbon dioxide radical, and carbonate radical, etc. Whereas examples of non-radicals are hydrogen peroxide, hypobromous acid, hypochlorous acid, ozone, organic peroxides, peroxynitrite, peroxynitrate, peroxynitrous acid, peroxomonocarbonate, nitric oxide, and hypochlorite, etc. Among these ROS elements, hydrogen peroxide and the superoxide anion radicals are key redox signaling agents. Now, what are free radicals? Free radicals are highly unstable and chemically reactive molecular species containing at least one unpaired electron in valence shell or outer orbit. They may be atom or molecule or ion. Instability or reactivity are gained due to the unpaired electron present in the valence shell or outer orbit. In order to gain stability, they can either donate or accept an electron from other molecules. Therefore, they can behave as reductants or oxidants respectively. Now, if you see this figure, in that case, free radical can steal electron from a stable atom to pair with its unpaired electron. In that condition, free radical will be converted to a stable molecule, stable atom, whereas the stable atom will be converted to an unstable atom or free radical. Another way, this unpaired electron can be free radical can donate unpaired electron to this stable atom. In that case also, stable atom can be converted to unstable and free radical is converted to a stable atom. Antioxidants donate an electron to the free radicals to stabilize that free radical. All the free radicals are not reactive oxygens, whereas all the reactive oxygens are not free radicals. Few free radicals are coming under reactive oxygen. So antioxidants are compounds which reduce, neutralize and prevent the damage of cells caused by free radicals and thus reduce or eliminate the risk of certain diseases. These compounds are often reducing agents. Reactive oxygen species elements are well known for playing a dual role as constructive and destructive species. Indeed, ROS are engaged in many redox governing activities of the cells to maintain cellular homeostasis. The rapid production of hydrogen peroxide and its precursor superoxide anion plays an important role in plant defense responses to pathogen attack. They play important roles in modulation of cell survival, cell death, cell differentiation, cell signaling, and inflammatory related factor production. However, its overproduction has been reported to result in oxidative stress, which is considered as a deleterious process and is involved in the damage of cell structures that causes various disease states. Different sources of ROS can be divided into two groups, internal sources and external sources. If you consider about the internal sources, that means ROS production inside the cell. ROS are common outcome of normal cellular metabolism of oxygen due to the leakage of electrons from the electron transport chain and by various enzyme activities of oxidoreductase enzymes, including NADPase oxidase, xanthine oxidase, myeloperoxidase, cytochrome P450 enzymes, glucose oxidase, lipo and cyclooxygenase, etc. Among them, NADPH oxidase represents a non-mitochondrial source of ROS generation and plays important role in superoxide formation via oxygen reduction mediated by electron donor NADPH. Different organelles are involved for the ROS production inside cells. Those are peroxisome, mitochondria, endoplasmic reticulums, and different growth factors including cytokines and chemokines are also involved for the production of ROS inside the cells. Now, if you consider about the external sources, examples are ionizing radiation, UV radiation, smoking, environmental pollution, fast food, stress, pesticides and insecticides, etc. They are also accelerating production of ROAs inside the cell. There are mainly two types of antioxidative defense system mechanisms present in cell to scavenge ROS, namely enzymatic and non-enzymatic defense systems. Enzymatic defense system means primary defense against ROS. In that case, mainly three enzymes are involved: superoxide dismutase or SOD, catalase, and peroxidase enzymes. Whereas if you consider about non-enzymatic defense system, in that case it is a secondary defense against ROS. Dietary antioxidants are coming under this group. Examples are ascorbate, tocopherol, carotenoids, reduced glutathione or GSH, then phenolic compounds, etc. Let's talk about enzymatic defense mechanism against ROS. In that case, already I told you three main enzymes are involved: superoxide dismutase, catalase, and peroxidase enzymes. The superoxide anion released during different cellular processes like oxidative phosphorylation process is first transformed to hydrogen peroxide with the help of superoxide dismutase and then further reduced to give water by catalase and peroxidase enzymes. In that case, glutathione is oxidized to GSSG. GSH is the reduced form and GSSG is the oxidized form. So during conversion of H2O to water, that process is actually reduction process, and therefore GSH, which is present in reduced form, it is actually oxidized during this conversion to GSSG. Superoxide dismutase is a metalloprotein present in both prokaryotic system and in eukaryotic system. In prokaryotic cells, it is iron containing and manganese containing. Whereas if you consider about eukaryotic systems, it is present in cytosol and as copper containing and zinc containing. Whereas in mitochondrial matrix as manganese containing SOD. It catalyzes the dismutation of superoxide anion into oxygen and hydrogen peroxide. It is first enzyme involved in the antioxidant defense mechanism. Second of all, glutathione peroxidase, which is a selenoenzyme. It is present in extracellular part or in cytoplasm, nucleus, mitochondria, endoplasmic reticulum. It catalyzes the reduction of hydrogen peroxide into water Simultaneously, glutathione is oxidized to glutathione disulfide. This enzyme is specific for GSH, that means glutathione reduced form, and non-specific for the hydroperoxides like H2O2 and lipid peroxides. Heme peroxidases. Heme peroxidases means it is heme containing enzyme present in bacteria, fungi, plants and animals. Examples are Horseradius peroxidases, lactoperoxidase and other mammalian peroxidases. It catalyzes the oxidation of various electron donors with the help of H2O2 and scavenges the endogenous H2O2. It converts H2O2 to water. Catalase, it is a heme enzyme. It present in peroxisome or microperoxisomes and cytoplasm of almost all mammalian cells. It converts H2O2 to water and oxygen. So these are the enzymatic defense mechanisms present inside cell. Let's talk about non-enzymatic defense mechanism or secondary defense against ROS. They are commonly consumed as dietary supplements. These are low molecular weight compounds like ascorbic acid, tocopherol, carotenoids, phenolic compounds, and reduced glutathione. These molecules react with free radicals to produce a lesser harmful radical species, hence they are antioxidant molecules. In membrane, alpha tocopherol is acting as the antioxidant and it can scavenge free radicals in the membrane, whereas ascorbate, phenolics and GSAs, they are present in the cytosol to scavenge ROS molecules. So this is overall cellular defense mechanism against ROS, namely enzymatic defense and non-enzymatic defense mechanisms. If ROS reaches above threshold level and if inbuilt defense mechanism of body unable to remove those toxic ROS properly, then oxidative stress occurs which leads to cell damage. Under severe oxidative stress condition, excess ROS damages biomolecules like DNA, protein, lipid, and to some extent carbohydrates. And significant molecular and physiological damages can take place ultimately, which lead to cell death or numerous diseased conditions. So harmful or destructive effects of ROS means oxidative damages of biomolecules, including lipid, protein, DNA, and few cases carbohydrates, which leads to uh, various disease conditions. ROS elements induce oxidative damage of both nuclear DNA and mitochondrial DNA. The nature of these damages are various modifications of bases and nucleotides, mostly in sequences with high guanosine because guanine is more susceptible to oxidative stress. Two main modifications of guanosine are 8-hydroxy-2'-deoxyguanosine or 8-oxo-7-8-dihydro-2'-deoxyguanosine. Other damages are strand breakage, oxidation of deoxyribose, removal of nucleotides and DNA protein cross-linking, etc. Hydroxyl radical produces different compounds from the DNA bases like 2,6-diamino-4-hydroxy, 5-formamino-dipyrimidine, 8-OH adenine, 2-OH adenine, thymine glycol, cysteine glycol, hydroxymethylurea, etc. The oxidative damage of mitochondrial DNA produce some abnormal compounds in the electron transport chain, thus more reactive oxygen species are generated due to increased leakage of electrons that promote uh, cancer and aging. Let's talk about oxidative damage of protein. Proteins can be modified by direct ways or by indirect ways. Activity of proteins can be modified directly by carbonylation, nitrosylation, disulfide bond formation, and glutathionylation. Whereas indirect ways means indirect modification of protein occurs by reaction with secondary byproducts of oxidative stress like byproducts of fatty acid peroxidation reactions. Oxidative changes Oxidative changes to proteins by ROS can lead to a number of consequences like site-specific amino acid modifications, peptide chain fragmentations, aggregation of cross-linked reaction products, increased susceptibility of proteins to proteolysis, alteration of electric charges, altered immunogenicity, increased or decreased uptake by cells, etc. ROS elements can also attack lipid molecules, which process is known as the lipid peroxidation process. That means it is a free radical chain reaction where free radicals steal electrons from the membrane lipids, either a cell membrane or organelle membrane, producing lipid peroxidation products, which further attack DNA, protein, and other lipid molecules, ultimately leading to cell death. ROS elements attack mainly on two common sites: phospholipid molecules having unsaturated bond between two carbon atoms and the ester linkage between glycerol and the fatty acid. Mainly two steps are involved in lipid peroxidation process: initiation, propagation, and termination. The initiation step involves production of a lipid radical when a reactive oxygen species like hydroxyl radical combines with a hydrogen atom of lipid, and it will produce lipid radical along with water. The unstable lipid radical quickly reacts with oxygen in the propagation step to form peroxyl lipid radical. Then this peroxyl lipid radical can react with lipid to produce lipid hydroperoxide. The chain reaction of free radical reactions continues until a non-radical species is formed by combination of two free radicals in the termination step. The free radical reactions can also be halted by antioxidants within an organism. They can bind to the free radicals and prevent lipid peroxidation process. The lipid molecules can also be oxidized or catalyzed by several enzymes like lipoxygenase, cyclooxygenase, and cytochrome P450. End products of lipid peroxidation process may be mutagenic or carcinogenic. The main primary products of lipid peroxidation are lipid hydroperoxide LWH. Among the many different aldehydes which can be formed as secondary products during lipid peroxidation are malonaldehyde MDA, propanol, hexanol, and 4-hydroxy nonenal, that means 4-HNE. MDA is most mutagenic product of lipid peroxidation, commonly used as a biomarker for oxidative deterioration in omega-3 and omega-6 fatty acids, whereas 4-HNE is the most toxic product and it displays a dual role as a protective signaling molecule during gene expression and a cytotoxic promoter of pathological pathways. This figure is showing you initiation, propagation, and termination pathways during peroxidation process of polyunsaturated fatty acid. Most often, reactive oxygen elements affect polyunsaturated fatty acids or PUFA because reactive hydrogen-containing methylene groups are present in between multiple double bonds. So, in this figure, two double bonds are present here, and this is the active methylene group where hydrogen is where reactive hydrogen is present. Now, except O2 minus and H2O2, most of the ROS elements can initiate this lipid peroxidation process. So, during initiation process means it is initiated by ROS elements. I have given one example that is the hydroxyl radical, and PUFA is converted to lipid radical. During that process, water is released, and this is the initiation process. After that, there will be molecular rearrangement of lipid radical, and so in this process, conjugated diene can be produced. Now, in the second step, that means in the propagation step, oxygen is reacted with the lipid radical so that lipid peroxyl radical is produced. Now, this lipid peroxyl radical is again reacting with PUFA, that means polyunsaturated fatty acid, to produce lipid hydroperoxide, and simultaneously PUFA is again converted to lipid radical. So this is about the propagation processes. In the last step, that means in the termination step, in the termination reaction, antioxidants donate hydrogen atom to the lipid peroxide radical and resulting in the formation of non-radical products. I hope already you have understood that the generation of free radicals causes no problem as long as there remains a balance between ROS production and neutralization by natural antioxidant defense mechanisms. But under severe oxidative stress conditions, if the cellular antioxidant defense systems become weak or the level of ROS reaches abnormally high, cells are damaged, which ultimately lead to the numerous pathological conditions, that means numerous diseases. Examples of diseases caused by ROS are arthritis, cardiovascular disease, hemorrhagic shock, cystic fibrosis, metabolic disorders, gastrointestinal ulcerogenesis, Alzheimer's disease, Parkinson's disease, apoptosis, necrosis, AIDS, myocardial ischemia, reperfusion, injury and aging. So, intake of huge amount of antioxidant rich food is very essential to combat the destructive effect of ROS. So this is overall summary of today's discussion. I have discussed about sources of ROS, defense against ROS, roles of ROS, and various diseases caused by ROS elements. Thank you very much for watching this video. Kindly like, share and subscribe this channel.